Here is an experiment. You are watching human stem cells dividing and dividing and dividing. So far as we know today, these are the only cells in the world that can keep dividing and dividing and dividing indefinitely. The only cells in the world, in other words, that are immortal. The dividing stem cells you are watching here are from the laboratory of James Thomson, a professor at the University of Wisconsin. It was here in this laboratory in Madison, Wisconsin, that Thomson and his colleagues first discovered a way to culture stem cells and to get pictures like these. And it is here at what is called the Y Cell Institute that 5 of the most fruitful stem cell lines in the world are now being cultured and distributed for worldwide research. Well, what exactly is a stem cell? Dr. Timothy Mulcahy, one of James Thomson's colleagues, explains.
Well, I think the simplest way to explain that is to think of stem cells as pretty much a blank cell. It's a cell that has the potential to do anything that any cell in the body does, but it hasn't received specific instructions yet as to which particular program to follow. So the beauty of stem cells and the potential that they offer for medicine, for example, is if we can understand the signals that'll tell the cells which programs to follow, we could then develop cells, for example, to replace cells that are injured in heart attacks, or we can make neural cells that could replace deficits that people suffer in Parkinson's disease. And unlike any other cell in the body, which is already following a preordained program, these cells are sitting there waiting to be told what to do. And if we can understand the science of how to do that, we have a very powerful tool to treat disease.
Where do stem cells come from? Well, there are 2 kinds of stem cells. One type is embryonic stem cells, and the other is adult stem cells. Let's look first at embryonic stem cells. Embryonic stem cells, as the name implies, come from a very early stage of the human embryo. All human embryos begin with a male sperm uniting with a female egg. This fertilization usually happens inside the mother's body. However, in modern in vitro fertilization clinics, couples who for one reason or another cannot conceive normally allow their sperm and egg to unite in a laboratory petri dish. The fertilized eggs in the laboratory are given the right nutrients and allowed to grow to a very early stage and then implanted in the uterus of a potential mother, where they grow to term and are born as healthy babies. In vitro fertilization, there are usually more embryos formed than the couple can or want to use, and the unused ones are frozen or destroyed. Today, some of these unused embryos are being made available, with the consent of the donors, to scientists like James Thomson who want to study stem cells. Well, a fertilized egg in the right environment begins to divide. 1 cell divides to become 2 cells. 2 divide to become 4. 4 become 8, 8 16, 16 32, 32 64, 64 128. At this point, 5 to 7 days after fertilization, the embryo is still a clump of cells, embryonic stem cells we call them. Each of these 100 or so stem cells has the potential to further divide and then differentiate to form blood cells, muscle cells, bone cells, nerve cells, skin cells, eventually to form the trillions of cells that make up a human baby. At this very early stage, however, this is only a potential. As yet, the embryo has no differentiated cells, no tissues, no organs, no feelings, no consciousness, no experience. What Thomson and other researchers around the world have done is to take some of these very early stem cells before they have begun to differentiate, separate them out, and culture them in the laboratory.
Unlike a lot of cell lines that we grow routinely in the lab, and we know how to grow very well, these cells are very finicky, and we still don't completely understand all the nutrient requirements, for example, that they have, so we can't easily grow them. So one of Jamie's major accomplishments was discovering a system which would allow us to grow them in the laboratory. So it is very labor intensive in the sense that you have to watch the cells very carefully. You have to be very scrupulous in what you provide in terms of media and nutrients for the cells. And you have to make sure that they don't begin to differentiate. And that's one of the tricks here. These cells, because they are kind of a blank slate to start with, if they receive a signal to tell them to start going down a certain pathway, they will go down that pathway. And a lot of what we want initially in the laboratory, and a lot of what Jamie was interested in, was studying the cells in their very earliest stages, before they had any program to follow.
One big difference between stem cells and all other cells of the body is that all other cells have an internal clock. That is, they only divide a set number of times before they die off. Some years ago, a researcher named Leonard Hayflick discovered what that set number was. It's about 50. That is, human body cells seem to reproduce about 50 times, and then that's it. They die of old age. And this is why one of the reasons that you too, barring accidents or disease, eventually will die of old age.
Stem cells are such that their biology allows them to continue to grow, as far as we can tell, indefinitely. And so that gives us two opportunities. One, we can have a long period of time to study the cells. Two, we can get large numbers of cells, which are going to be necessary for any therapeutic application. So the real trick, and we don't quite understand why these cells don't start their biological clock. Their clock is set at 0 and hasn't started timing. And again, that's one of the most incredible features.
The hope is that researchers around the world will discover the right signals that will make stem cells turn into healthy muscle cells, or blood cells, or nerve cells. And there have been some successes already. Dan Kaufman and his colleagues at the Y-Cell Institute discovered ways to signal stem cells to develop into the blood cells you see in this photo through the microscope. In November of 2001, Dr. Su-Chun Zhang and his colleagues at Y-Cell Institute produced stem cell-derived neurons. These stem cells were successfully transplanted into the brains of newborn mice. The neuron that we're seeing after transplant is almost identical to what the neuron should be in the healthy brain, reported Dr. Zhang. These transplanted cells had no experience in the brain, and we wanted to see if they would mirror the development of the mouse brain, and they do. These are the cells that will be used ultimately to treat Parkinson's and other central nervous system disorders. Kierstead was also one of the
Now, there are other ways to obtain embryos for the purpose of deriving stem cells. One is to use an in vitro fertilization lab to simply make an embryo specifically to retrieve its stem cells. That was done in Norfolk, Virginia earlier this year and announced, and generated a fair amount of controversy. And the second is to use cloning technologies to make embryos from which stem cells can be derived.
When Dr. Chero explained this technique of therapeutic cloning in 2002, she added that, quote, most scientists think this is fairly far down the road, unquote. You know, it turned out that just a few years later, a team of Korean scientists did just that. In 2005, they created nearly a dozen new embryonic cell lines using therapeutic cloning techniques. They started by taking skin cells of diseased or injured patients. Using a technique similar to the one that created the first cloned mammal, Dolly, they took the nuclear DNA from the patient's skin cell, inserted it into a human oocyte, egg cell, that had its original nucleus removed. Then they chemically stimulated the oocyte with its new DNA until it began to divide. After approximately 6 days, just long enough to derive some stem cells, but long before the oocyte would develop into an actual human embryo, they harvested the stem cells produced and let them reproduce indefinitely. Since these stem cells have the exact genetic information of the original diseased or injured donors, their hope is they will prove to be therapeutically useful in curing the diseases or repairing the injuries of the donors. Most of the ethical objections to stem cell research come from people who feel strongly that the human embryo, even at its earliest stages, is a human person and is entitled to the same protections that all human beings have, whatever their age, abilities, or disabilities.
The debate about what constitutes morally significant forms of life is one that has gone on for centuries and is not likely to be resolved anytime in our lifetimes. At base, it touches on things that cannot be proved and things that cannot be explored through experimentation. For example, whether you think that the essence of moral significance lies in potentiality, or you think the essence of moral significance lies in a kind of an experiential view of life. That is, if you believe that the mere potential to develop under the right circumstances into a baby means that this form of life must be protected. In other words, the acorn should be protected as if it were already an oak. Then you're making this value judgment based upon a vision of the world that kind of transcends time, in which we see life as part of a continuum across time. On the other hand, there are people for whom that is not a relevant factor, and they ask only, do we have an entity that can experience itself and can feel disappointment, pain, in other words, can be harmed? And they look at an embryo and say, no, it doesn't even have the biological substrates to be self-aware, to have formed a desire to continue to exist. And so cutting off its existence in no way thwarts its desires or harms it. And these are very different views about moral significance, and they can't be brought together.
How can we as a society then decide such irreconcilable differences? Should we simply put them up to a vote and let majority rule?
No.
Well, I personally believe that there's actually a lot of guidance within the philosophy of our Constitution. The United States is not governed by pure popular majority. Instead, we have a mix. On topics that encompass rather ordinary sets of concerns, we allow the popular will to prevail. Should people be allowed to drink or drive or vote or drink or drive at 18 or at 20 or at 21, and we put that to a popular vote. On the other hand, there are areas of life that we have said are so fundamental, so central to our personal identities or to our civic life, that even when the popular sentiment would suggest that these activities should be restricted, we will permit them until the most compelling argument has been made for restriction and until we have been shown that there's no other way to handle this concern other than restricting the activity. Those are things that are listed in the Bill of Rights, like the freedom of association, of speech, of practicing your own religion. And it's also a set of things that have been identified by the Supreme Court as implicit within the Constitution and the Bill of Rights, such as the freedom to marry. Now, one of the areas that we need to examine is the freedom of scientific inquiry. There have been suggestions over the years that this is something akin to free speech, that just as free speech ensures the long-term stability of a civic society by providing an outlet for dissent, something other than violent revolution, does scientific inquiry, by ensuring the continued development of new knowledge, help to stabilize society or destabilize society? In my mind, that's a very important question, because if we see it as fundamental to the long-term stability of a civic society, then it's something that needs to be protected even against the popular will.
What about the future? What will happen 10 to 20 years from now with stem cell research? In April of 2005, the National Academy of Sciences proposed new ethical guidelines for research on embryonic stem cell lines that attempt to bridge some of the differences between religious and scientific points of view. The new guidelines oppose reproductive cloning, but support therapeutic cloning. They pave the way for research projects that inject human stem cells into some animals but oppose injecting them into non-human primates like monkeys or apes. They advise that human embryos should not be grown in culture for more than 14 days, the time when the first hints of a human nervous system begin to appear, and they recommend that human egg donors not be paid. At present, the legal status of stem cell research in the United States is a mixed story. Some states have passed laws to ban both reproductive and therapeutic cloning for stem cell research. Other states are considering such bans. On the other hand, some states, like California, have passed popularly supported referendums to give generous support to stem cell research in all its variations. On the federal level, there are no laws at present restricting stem cell research. The U.S. government, however, has ordered the National Institutes of Health to refuse federal funding for any stem cell research using embryonic stem cell lines created after August 9, 2001. This restriction is being contested by many scientists, as well as many scientific, religious, and political groups. There are similar conflicts about the ethical and legal status of stem cell research in Western European countries. On the other hand, countries in Asia, like China, South Korea, Singapore, and Taiwan, place no restrictions on embryonic stem cell research, and scientists there are making rapid progress in therapeutic cloning, as well as many other frontiers of genetic engineering and cutting-edge biology. How fast will progress take us in the 21st century? No one can be sure. Dr. Mulcahy gives voice to the majority view of scientists today.
I would say 10 to 20 years from now, I fully expect that we'll have a number of effective treatments based on embryonic stem cells. I also would predict that much like what happened in the case of in vitro fertilization, which at its inception was a real hot button, it will evolve into a much more accepted and appreciated medical tool.
Just as Dr. Mulcahy pointed to a change of public attitudes about in vitro fertilization over a few decades, Dr. Cherrill pointed to a similar change in other controversial areas of the past.
When the first stem cell announcement came out from James Thomson's lab, we saw the very same people who had opposed federal financing on embryos back in '93 suddenly rethinking their opinions and saying, well, you know, we've got these large numbers of people with terrible diseases like Parkinson's and diabetes, and we're all facing heart disease, and am I really willing to sacrifice that? Well, maybe not for the very earliest embryo. Maybe my concern's really more about more advanced forms of fetal life. And we saw shifting. But of course, cloning is a terrible thing, and I don't like cloning in any fashion. Then, at the end of 2001, we begin to hear more specific discussion about real medical advances that might be made possible by using cloning just to copy a cell and make it divide a few times, but not to make it into a baby. And the very people who were most adamantly opposed to cloning are beginning to separate their opinions into an opposition maybe to reproductive cloning, but a tolerance for research cloning.
And indeed, that research continues today. In cloning, genomes, and on stem cells. And the consequences for the 21st century, for the third millennium, are still to be determined.